this video, we're going to be talking about the liver. So we'll start off with the function and location of the liver itself. So the liver is located on the lower side of the rib cage on the right side. So the function of the liver is to maintain blood glucose levels, and it maintains iron homeostasis as well. It also distributes cholesterol and manufactures plasma proteins. Just to reiterate that one more time, the function is to maintain blood glucose and iron homeostasis, as well as distributing cholesterol and manufacturing plasma proteins. And the location of the liver is on the lower side of the ribcage on the right side. Moving on, we're going to be talking about the blood supply, as the liver is actually connected to blood supply. I'm going to start off with drawing a diagram. I'll start off by drawing the heart. For the sake of an example, just to make it really easy to understand, this triangle here is the heart, easy to see. Circle here is the liver, and this green rectangle is the intestine. Now we're going to talk about the direction of blood supply and the specific veins and arteries responsible for transporting blood to specific locations. So we'll start off by explaining the hepatic portal vein. So the hepatic portal vein takes blood from the intestine to the liver, and this is blood low in oxygen, and it will have a high amount of nutrients if food was eaten because it's coming from the intestine. So the hepatic portal vein takes food from the intestine to the liver, as you can see here. Great. So that's the hepatic portal vein, transporting the deoxygenated blood from the intestine to the liver. But this blood will likely have a lot of nutrients, but it's also possible that it will have bacteria contamination, and the liver will help destroy that bacteria contamination. And this, interestingly enough, this actually accounts for 75% of the blood that flows into the liver. Now, the hepatic vein transports blood from the liver to the heart, and this blood is again low in oxygen. Now, the hepatic artery transports oxygen-rich blood from the heart to the liver, and this blood will have a high level of nutrients, and these nutrients have been filtered by the liver. So that's going from the heart to the liver. Yeah. And importantly, just to understand, a vessel is actually also present that transports blood from the heart to the intestine, and this will be oxygen-rich blood again. So I'll just draw that here. Okay, great. That's just a little diagram of how blood is transported from the heart to the liver and from the liver to the intestine, and how everything works together to facilitate the efficient transport of blood. Added in dot points here. 75% of blood that flows into the liver is from the hepatic portal vein. The hepatic vein will transport deoxygenated blood again. And this is blood that's going from the liver to the heart. And the hepatic artery going from the heart to the liver transports oxygen-rich blood. Okay, great. So yeah, hopefully that all makes sense, and hopefully this diagram just helps you understand how blood is transported. Moving on, we're actually going to be talking about the internal anatomy of the liver. So just some key things to understand about the internal anatomy is that hepatocytes are liver cells. They're the main cells of the liver, and these cells are arranged into hexagonal structures. So we're actually going to draw a hepatocyte now. This is just a really rough drawing of a hepatocyte. There's specific regions of the hepatocyte which are important to understand. I'm going to draw these in first, and then we'll go through and explain what their specific function actually is. So there's these little sections here, and these are called portal triads. Another important part of the hepatocyte that I want you to know is the central vein. So blood will actually leave the portal triad. It'll move across the tissue towards the central vein, and it'll leave the liver, and as the blood moves through this tissue, all the functions of the liver are enacted upon the blood. So blood will leave these portal triads, these little circular structures on the edges of the hepatocyte, and it'll move across the tissue towards the central vein, which is in the center of these hepatocytes, and it'll leave the liver. And as the blood moves through the tissue, all the functions are enacted upon the blood by the liver. Okay, so portal triads are composed of 3 small vessels. They're composed of the small branch of the hepatic artery, a small branch of the portal vein, and a section of the hepatic duct. And the hepatic duct essentially conducts bile made in the liver away from liver tissue. Small branch of hepatic artery, small branch of portal vein. Okay, great. So that's just an overview of the internal anatomy of the liver and liver cells and portal triads and their specific structure that allows them to facilitate the transport of blood out of the liver. So moving on, we're going to be talking about the process of glucose homeostasis, because the liver actually plays a really important role in regulating blood glucose levels. Glucagon causes the liver to release stored glucose, and insulin causes the liver to store glucose as glycogen. Okay, and now we're going to be talking about cholesterol balance, because the liver plays a really important role in that. So cholesterol keeps all cell membranes flexible, and it forms the basis of testosterone and progesterone. So cholesterol does not mix easily with blood and water, and it can cause blockages. I'm just going to write it down. So yeah, this is just a quick background on cholesterol. It keeps membranes flexible and forms the basis of testosterone and progesterone. However, it does not mix easily with blood and water, and that's an important thing to understand. The liver actually produces high density and low density lipoproteins. We'll go over each specific one now. Lipoproteins. So high density lipoproteins transport cholesterol away from peripheral vessels and back towards the liver, and they're termed as good cholesterol as it reduces the size of cholesterol deposits that can cause blockages. And then we have low density lipoproteins, and low density lipoproteins are considered bad cholesterol because it transports cholesterol from the liver to deposits, making these deposits bigger and potentially causing blockages. Great, so that's the function of high density and low density lipoproteins, and just an overview of cholesterol and its important role in the body. The liver actually makes its own cholesterol through conversion of triglycerides, and it's capable of removing cholesterol when it conducts bile. Now we're going to be talking about another really important process, the function of an insoluble protein, albumin. So albumin is a protein that the liver makes that is released into the bloodstream to maintain blood osmolality. Albumin sits in the bloodstream and is a soluble protein that increases the amount of dissolved material in the bloodstream, which helps to raise the solute concentration in the blood to make it more equal to the solute concentration outside of blood cells, and this prevents water from leaving blood vessels via osmosis to the extracellular fluid, as water always moves from low to high solute concentration via osmosis. I'm going to draw a diagram because that may seem like a lot of information. Okay, so let's say this is a blood vessel here, and all these little circles that I'm drawing are blood cells. So red blood cells are all here, and albumin, I'm going to draw in a purple color, is also here. Let's say that this region below, so this region here, is a very low osmolality region. However, the region above is very high in osmolality. This albumin, these little purple circles, will sit in the bloodstream, and it'll increase the amount of dissolved material in the bloodstream. And this increases the amount of solute concentration in the blood to make it more equal to the solute concentration outside of the blood, so outside this section here. And this prevents water from leaving the blood vessel via osmosis to the extracellular fluid, because water always moves from a low to high solute concentration via the process of osmosis. Okay, so that's just something to understand about albumin and its unique function in preventing water loss out of blood vessels. Moving on, we're going to be talking about iron balance, because the liver plays a really important role in iron homeostasis. So iron helps red blood cells to carry oxygen, and red blood cells contain hemoglobin, which attracts oxygen and takes it to the cells where it is needed. However, it needs iron to do this. Red blood cell is here. Within the red blood cell itself, there is a hemoglobin. So this is the square that I'm drawing here. And within this hemoglobin, there is an iron. I'm going to say Fe. So there's this red blood cell which contains hemoglobin, the protein hemoglobin, and this attracts oxygen and takes it to the cells where it's needed, and it needs iron to do this. And iron is stored within that hemoglobin protein. Now, in terms of transporting iron in general, iron is actually transported via transferrin, the glycoprotein, which is a glycoprotein. This carries 2 atoms of iron at a time. Now, in terms of iron storage, iron is actually stored in a structure called ferritin. And the liver releases its stores of iron stored within ferritin into the bloodstream. This is an important thing to understand as well. How does iron actually get in and out of cells? So iron is bound to this transferrin, and transferrin will bind to a transferrin receptor, and this receptor allows it to enter a cell. So iron is bound to transferrin. Transferrin will bind to a... So just to reiterate that one more time, iron is transported via transferrin, a glycoprotein. It's stored in a storage protein called ferritin. It gets in and out of cells as when it's bound to transferrin, a transferrin molecule will bind to a transferrin receptor, and this allows iron to enter the cell. And iron leaves the cell via a transmembrane iron transporter called ferroportin. So ferroportin is found in gut cells, white blood cells, the liver. Essentially what it does is it facilitates the process of iron leaving the cell. Ferroportin essentially will act like a tap. It releases iron into the bloodstream. Normal levels of transferrin are controlled by the hormone hepcidin, and hepcidin essentially turns the tap off in a way. It binds to ferroportin, removes it from the membrane, preventing the release of iron from cells into the bloodstream. The structures of ferroportin, the transmembrane iron transporter, acts like a tap, releasing stored iron into the bloodstream, and the hormone hepcidin prevents this ferroportin from releasing stored iron, and these two processes help maintain iron homeostasis within the body. Hepcidin is made by the liver. It is released in moderate levels under normal conditions, but when the cells of the liver detect that iron content in blood is too high, the hepcidin release will begin, and it'll act on the liver cells, gut cells, white blood cells to prevent these cells from releasing stored iron. Now, if the iron circulation is too low, then hepcidin release will decline to allow for, say, the transmembrane transporter ferroportin to release stored iron into the bloodstream to increase iron levels within the blood. Say if you have a stimulus of consuming red meat, the variable will result in an increased iron level within the body, and the receptor, the transferrin receptor on the hepatocyte cell surface, will facilitate the control center, which in this case is the hepatocyte, to produce hepcidin. Hepcidin will act on the ferroportin channels and turn the tap off. It'll bind to these ferroportins, removing it from the membrane and prevent the release of iron from cells into the bloodstream to help maintain iron homeostasis. Okay, that is the video on the liver. This was definitely a lot of content that we covered in this video. Hopefully you learned a little bit more about the liver and the specific processes that occur within it. And making this video definitely helped me solidify my knowledge in these processes and these specific topics. Thanks for watching, and that's all we're going to be covering in this video.